So next is Hershey-Chase experiment. So this Hershey-Chase experiment was also conducted or demonstrated to prove DNA is the genetic material. Why this experiment was conducted is even after Griffith's experiment and Avery and others experiment, still some scientists believed that protein and not DNA was the hereditary material. Because if you see an eukaryotic cell, there are equal amount of protein and DNA. So they thought that this protein has also a role in transforming the genetic material. Hershey and Chase finally they provided a convincing evidence that DNA is the genetic material, and this experiment was conducted in the year 1952. For this they selected T2 bacteriophage. So protein codes of phages are radioactively labeled here. So here you can see the protein codes are labeled here. Here DNA of phage is radioactively labeled. So here the protein, that is outer covering alone, is labeled here. In this T2, this inside DNA is radioactively labeled. So this is contains radioactively labeled phosphorus and this contains radioactively labeled sulfur, because sulfur is present in the protein code and phosphorus present in the DNA. The phages, they infect the particular E. coli bacteria. Phages infect bacteria with the genetic material. You know this when the phage that is when the bacteriophage affects the bacteria, only the genetic material goes inside. This you have learned in 11th standard. Only the genetic material goes inside, whereas the remaining protein coat is left outside and it is called as ghost. Do you all remember that? So the outer coat, protein coat, is called as ghost, which is left outside. Only the genetic material enters inside. So phage infect bacteria with genetic material. In both the cases you can see phages infect bacteria with the genetic material. Third one, no radioactivity enters the cell. Why? Because the radioactivity is present only outside. What has entered inside is only DNA. So no radioactivity was found in this cell, but here radioactivity enters the cell because what is entered here is DNA, because the radioactivity is present in the DNA here. So this is what is proved by Hershey and Chase in the experiment. This the same you can see in the video. Now we will see a video about Hershey and Chase experiment to prove that DNA is the genetic material. In 1953, Alfred Hershey and Martha Chase confirmed DNA's predominant role in genetics by demonstrating that DNA is the genetic material of a virus called phage T2. The phage, which infects E. coli, consists of a head, sheath, tail and a basal plate made of different proteins. DNA is packaged within the head of the virus, and T2 comes in contact with E. coli. The phage attaches to the bacterium by its tail. Next, the phage injects the genetic material into the cell. The genetic material directs bacterial enzyme to produce viral offsprings. When the life cycle is complete, 100 to 200 progeny phages have been assembled inside each bacterium. The bacterium breaks open or lyses and the phages are released. Since T2 consists of only DNA and protein, Hershey and Chase reasoned that the genetic material must be one of the two components. So they designed an experiment to determine which it is. Further experiment, the scientists prepared T2 phages that either had radioactive DNA or radioactive protein. To make the components radioactive, Hershey and Chase infected E. coli with T2 and grew the bacterial cells in two different culture mediums, one containing radioactive isotope phosphorus 32 and the other containing radioactive isotope sulfur 35. All the phages released from the bacteria grown in phosphorus 32 medium had radioactively labeled DNA, while all the phages released from the bacterium grown in the sulfur 35 medium had radioactively labeled proteins. In the next phase of their experiment, Hershey and Chase infected E. coli with two types of labeled T2. They then used a blender and a centrifuge to separate the protein parts of the phage called the phage ghost from the rest of the infected cells. The scientists discovered that the labeled DNA appeared in the host cells but not the phage ghost, while the labeled proteins appeared in the phage ghost but not the host cells. In addition, the progeny of the T2 with labeled DNA were radioactive, while the progeny of the T2 with labeled proteins were not radioactive. As DNA contains phosphorus, the radioactive isotopes of phosphorus binds with DNA. As protein contains sulfur, radioactive sulfur binds with the protein coat of T2. So in the first case, phage ghost is non-radioactive. Phage ghost is the protein coat which is separated from the DNA. In the second case, E. coli is non-radioactive. So the progeny that comes out from the bacteria are also non-radioactive. If protein was the genetic material, in the second case also the E. coli would have been radioactive. As DNA is the genetic material, the first case released radioactive progeny phases. So this proves that DNA has entered inside. So that is why it was seen that E. coli was radioactive. The second one, it did not show radioactive because only the phage ghost shows radioactivity. So this proves that DNA is the genetic material. Because genetic material is passed on from parent to offspring, Hershey-Chase concluded that DNA had to be the genetic material of T2. The results have convinced the scientific community that DNA was the hereditary material. Today we have seen two important topics, Oswald and Avery experiment and Hershey-Chase experiment. These two are important 5-mark questions. Thank you, students.